link to a quick evaluation survey. Please fill it out for a few reasons. It gives us your feedback on how to refine and reshape these sessions to address your priorities. And filling out this survey also qualifies you for receiving free CME credits. Slide, Taylor. Thank you. And for those who may be new to ECHO, it stands for Extension for Community Health Outcomes. This is a model of lifelong learning and guided practice that connects community-based health workers with each other in a multidisciplinary team of subject matter experts to create a community of practice and learning. We are all learners and all teachers in this space. This is a space for you to share your own challenges, strategies, resources, and lessons learned with your peers and colleagues. And next slide. And I will pass the mic over to Dr. Stigler-Granados to continue the introduction. Thank you.
Thanks, Kato. Welcome, everybody. I am happy to see so many of you here this morning and this afternoon. We're in multiple time zones right now. This is a brief agenda. We're going to have some introductions where I'll introduce our speakers. And then we're going to have, you're in for a treat today, actually. This is going to be a great talk. We have Major Paul Lenhart here, and we also have retired Major Josh Bast with us. Then we'll have some time for Q&A. And then at the end, stay tuned because we'll need you to fill out an evaluation. So we'll have a link to the evaluation we'll post in the chat, and we'll also have a QR code for this at the end. Can I have next slide, please? And by the way, I am Dr. Paula Stigler-Granados. I am jointly affiliated with Texas State University and San Diego State University. I'm in both places. I'm actually in San Antonio right now, and I'll be going back to San Diego at the end of August. So I see lots of familiar faces today. And so I'm just excited to have you all here. I am a subject matter expert on Chagas disease and have invited our guest speakers today to talk with us. And I'm just super excited to have their expertise here because they are just rock stars in this field. So Major Paul Lenhart, I have had the honor of working with for several years now on a grant that we have through the Department of Defense, the Global Health Engagement Research Initiative. Quite a long biography here, but just a little bit about him. He's the Deputy Chief of Entomology at Public Health Command West at Joint Base San Antonio, Fort Sam Houston, at least I think until next Tuesday. And then he's taking another post. Major Lenhart got his BS in biology at UTEP and then PhD in entomology at Texas A&M. He also has had assignments, including operations officer for the 261st Multifunction Medical Battalion, Fort Bragg, North Carolina, and the executive officer for the 155th Medical Detachment and Preventative Medicine for Fort Bragg in North Carolina. Also, medical operations officer for 261st Multifunction Medical Battalion, Fort Bragg, North Carolina. Oh, that's in there twice, right? I just read that twice, but that's okay. He did it twice, maybe. He commissioned into the U.S. Army in June 2018. We were so lucky to have him. Prior to military service, he was postdoc scholar at the University of Kentucky Department of Entomology. He's authored 12 scientific pubs, given 41 conference presentations. He's had over 1.7 million in funding. He is extensively educated in entomology. Been involved in a lot of different research topics, including triatomies you're going to hear about today, tropical ant taxonomy, predatory bat diets. Major Lenhart, we're going to have to talk about that more. That's really cool. Facultative insect I can say that, biology. I apologize to my entomology colleagues out there. And predator-prey interactions. As an Army entomologist, he's studied medical arthropod threats with projects like kissing bugs, black flies, fleas, ticks, and mosquitoes. And here's his new post. He's going to be serving as the chief of U.S. Army's Walter Reed Biosystematics Unit based at the Smithsonian Institute. Next slide, please. And then also, I'm very excited to be working again with Josh Bast. He is currently serves the chief of the Program Development, Education, and Data Branch for the Defense Centers for Public Health at Aberdeen's Entomological Sciences Division in Edgewood, Maryland. In his current role, he leads a team that conducts commercial off-the-shelf product evaluations, develops educational materials, generates risk assessment products, and curates and analyzes vector surveillance, pathogen testing, and pest management data. He got his MS in entomology from Clemson University before he joined the Army as a medical entomologist in 2003. I had the honor of working with Josh on the same project that I worked with Major Lenhart on for multiple years before he retired. So it's really great to have him back on this project right now. co-presenting. He's held multiple positions of leadership in his 20-year career as an Army officer, including commanding preventative medicine detachment in combat while serving as senior force health protection consultant to U.S. forces in Afghanistan and leading 2 Army research laboratories in Kenya and the Republic of Georgia. In his 9 years executing DoD research and public health programs, he received 33 grants, totaling nearly $10 million. Way to go, Josh. I didn't know that about you. That's awesome. And he currently serves as the chair of the Armed Forces Pest Management Board's Risk Assessment Committee. So we're very, very honored to have him and his expertise here. We have 2 of the best. And so with that, no pressure, guys. I'm going to turn it over to you. Thank you both for your service, and also for all of you joining us from the military side of things. Thank you also for your service. And I will hand the floor over to Major Len Hart and Josh.
All right.
Thanks so much, Paula. I'll let Paul start us off.
Yeah, thank you, Paula. I really appreciate the very spirited introduction. I know we have a wide range of people attending, and I'm very thankful for all of you for being here this morning or afternoon, whichever it is for you. Obviously, we have a bunch of people who are providers, public health officials, veterinarians, and we're entomologists. So the focus we're going to give you today is on the bugs, but obviously with the One Health approach that we're all embracing, it's important for the vets and providers and public health officials to understand the bugs just as much as it's important for us to understand what happens after the bug bites you, or in this case, defecates on your face. And so, we all need to understand the different parts of the puzzle here to solve these zoonotic disease problems. A brief disclaimer here at the beginning. So any of the views or opinions that we present here are not necessarily those of the U.S. Department of Defense, and we may refer to some products, services, or websites that doesn't constitute an endorsement by the DoD of those products or services. As an added incentive, not just we're going to talk about cool bugs, but as an incentive for y'all to learn the material, I actually have a U.S. Army challenge coin, and we'll have a quiz later using the program Kahoot, or an app. You won't have to download the app. We'll give you a link a little bit later, and we'll do a quiz. And the winner of it, I want you to stick around afterwards. I'll get your contact info and ship you the coin. So our brief agenda, so we're going to talk briefly about the disease overview, about transmission and where the disease is. Then we'll go into the bugs, diversity and natural history, and then we'll move on to the military readiness. And finally, we'll have a case study exercise where we want everyone's involvement to kind of work through a problem set. Okay, here's an overview of the disease. And again, once my standard line a lot of times is that once the bug bites you, my job as the entomologist is over, or as a preventive medicine person. But obviously, it's not true. We need to understand what's going on after you get infected, and it obviously gives that gravitas to the importance of preventive medicine. So, Chagas disease is caused by a protozoan parasite, Trypanosoma cruzi. There's an estimated 7 million people infected worldwide. You'll see estimates widely on either side of that. But those infections occur primarily in the Americas. There are 2 phases to the disease, especially in humans. So in the initial acute phase, it lasts 2 months after infection. And this is when there's a high number of the parasites circulating in the blood. In most cases, symptoms are really absent. They're or mild, or if they are present, they're non-specific. So like fever, headache. You may get what's called a chagoma, which is a kind of a response to the infection site, and so you get like almost like a large bug bite. Swollen, painful, itchy. And if you're infected in the conjunctiva of the eye, you may get what's called Romagn's sign. So there's a picture of a child there with the Romagn's sign on the right. But again, these symptoms are usually very rare in the acute phase. During the chronic phase, the parasites are hidden mainly in the heart and digestive muscle. And then 1 to 3 decades after infection, up to a third of patients suffer from cardiac disorders, and up to 1 in 10 suffer from digestive, typically enlargement of the esophagus or the colon. And you may also get some neurological symptoms. So yeah, these symptoms come down as cardiomyopathy. The parasites they're damaging that tissue. They'll also get into the colon and esophagus. And they can result in eventual heart failure and sudden death. From the parasitologist perspective, here's the life cycles that we all love to see. So in humans, the parasite enters the body in a stage called the trypomastigote. They penetrate various cells and transform into amastigotes, and then this is where they start to multiply inside cells of the host. That it can then turn back into trypomastigotes and burst out of the cells, entering the bloodstream, and these cycles can occur multiple times in that infective stage. Then, if an uninfected bug takes a blood meal from the human or an animal with the parasite in the bloodstream, they become infected. The parasites replicate in the bug's gut in what's called the epimastigote stage, and then the cycle can repeat itself. The main factor we're leaving out that I left out here is the transmission stage, which you can see up here. So let's see what happens. How does that transmission actually happen? We like to talk about bug bites, but in this case, it's actually the feces of the bug that's the primary motive of infection. So contact with the feces of infected bugs is, again, primary method that Chagas is transmitted. Usually the infected patient scratched or smeared the parasite into the bite wound, into skin breaks, or the conjunctiva of the eyes or the mouth. It can also occur by ingestion of the infected bug. This is pretty rare in humans, but in our canines, this is probably the most common method of infection. Vector competency based on the species of the bug varies a bit. So there's been a lot of studies in the past, and you'll see ends up being more kind of, I think, more of a myth that the timing of the defecation is really important in transmission, and that species in more arid environments like the U.S. Southwest, won't transmit it because they're retaining water and they don't defecate. The issue is they can transmit it. They do. It's a better predictor, really, of the vector competency is how closely tied that species of bug is with humans. Surveillance efforts in the U.S. have found around half the bugs on average are infected. So depending on state, anywhere from 17 to 59% of the kissing bugs are infected, with Texas and Louisiana being the highest, New Mexico, Arizona, and California being the lowest. But there are other modes of transmission. So another common one is food and drink contamination with the parasite. So if you have a unprocessed juice, like fruit juices, where there was bugs contaminating the fruit when it was crushed, you might get infected that way. There's vertical transmission from mothers to their offspring during pregnancy or childbirth. Blood transfusions. This is actually the major way that we discover cases in the United States, is that we test blood products routinely for Chagas, at least the first time that a new donor is entered in the system. And that's how a lot of cases in the U.S. get diagnosed. And then there's organ transplants and laboratory accidents as well. Okay, moving on from transmission, let's look at range. So I've got a couple different visualizations here. In this figure, you can kind of get an idea of the extent of Chagas disease range in Latin America, as well as some in the United States, but some of this data is not accurate. So we definitely have the vector and the pathogen in the United States with endemic transmission. For instance, Texas is our main one. So that's not quite accurate there. This visualization here kind of shows the importance of what's called the Chaco region here in northern Argentina and Bolivia and Paraguay. So here we're seeing different important vector species around Latin America. You're seeing some different initiatives to combat them in the color-coded countries. These bars here are the infection prevalence and incidence of Trypanosoma cruzi, and then the colored circles here represent the kind of density of the vector population, and the color coding is red or yellow, shows some signs of insecticide resistance. So a lot of information there, but I want you to take away that this Chaco is really the kind of the epicenter for a lot of cases in Latin America. And again, here's another visualization showing different levels of kind of combating the pathogen and the vectors across different areas. So some geographic regions state that they've really interrupted the transmission by the main vector. Some of them have no programs instituted, and again here for the United States, no data included. But we'll see in a second that that's there's definitely local transmission. Okay, so here's a great map from Texas Department of State Health Services. So as you can see here, there are counties highlighted here that have locally acquired human cases, and this is not a new phenomenon. It's gone back, we have records going back quite a ways that there's been endemic transmission. There's also counties colored in here showing positive triatoma in bugs and positive animals. And there's a lot of cases of Chagas disease when we start looking at the wildlife in many of these regions. We look at the entire United States. Here are some of the really common triatoma species and their ranges. So I just focused here on Texas, but just recently it found Triatoma sanguisuga from Delaware infected with the parasite. So it is widespread in the United States, and it has been here. Okay, so let's talk a little bit more about the bugs themselves. Common names are kind of the bane of a lot of entomologists' existence. They vary by place to place. That's why we like Latin names. But depending on what country you are in the US or Latin America, there will be different nicknames for them. I'll be referring to them as kissing bugs. This is a group of insects that's in the subfamily Triatominae. We'll see in a minute what the different groups look like within there. They're in the family Reduviidae. These are the assassin bugs. Most of them are predators on other insects, and they use that proboscis to stab and suck up the juices from other insects. And all of these are within the order Hemiptera, which are all the true bugs. And there's a lot of plant-feeding insects here. There's a lot of omnivores. It's a very diverse group. So when we look at triatomans specifically, in terms of insects, they're not super diverse, 157 species, but for public health workers trying to identify these, it is a daunting task. There's 18 different known genera. Within the United States, we have 11 species. So most of them are triatoma, with 2 species in the paratriatoma. And there are some species in Asia, about 14 of them. None of them have been found to be infected with Trypanosoma cruzi, but they're in Southeast Asia and southern India, Philippines. And we have one pantropical species, Triatoma rubrifasciata. So this one is in the Americas, where it can vector Chagas disease, but it's also found in Africa and Asia as well, where it does not carry the disease yet. 3 of these genera, Triatoma rhodnius and Panstrongyllis, include most of our really important vector species. And these are the ones that are really can be tied specifically to living close to humans and our domestic animals. Here's some more visualizations kind of showing, different regions of Latin America in the bottom left have a different primary vector species. It's closely associated with humans. So Central America, we're looking at Triatoma dimidiata. There's also Rhodnius proxylus. Rhodnius is a genus of kind of like palm tree and palm thatch specialists. And then when we go down to the Chaco region, we're really talking about Triatoma infestans that just loves living with people. When we look at our United States species, these are the 11 in the upper right-hand corner, so you can kind of get an idea of the diversity in body forms there. And Triatoma gerstachii is probably the most likely to transmit the disease, but it's also in Protracta, Rubida, and Sanguisuga all over the eastern United States. And there's some great efforts to kind of combine surveillance and locality data. So this database DataTri is a kind of great resource for what species are where and kind of resolving the taxonomy that's changed over time. So what is the life cycle of these bugs? They are, again, true bugs and hemipterans. So they have what's called gradual metamorphosis. So they lay eggs. Here are the huevos. And then we have 5 different nymphal instars. So these are immature stages that get progressively larger. They have one molt in between until they become a mature adult that has wings and reproductive organs. You can see kind of the wing buds growing on these nymphs. So the 5th instar has these large wing pads that when it molts will expand into those wings and they'll fly. What's interesting about gradual metamorphosis is that it means that the insects are, as immatures, are feeding on the same resource usually and living in the same place as the adults. It's not like a butterfly, where like the caterpillars are feeding on leaves and the adults are feeding on nectar and they kind of partition those resources. No, here they're all sucking blood. So let's talk a little bit more about natural history. So the primary hosts, at least especially in the United States, are wood rats in the genus Neotoma. But they feed on a wide variety of mammals, reptiles, amphibians, and birds. Like there you can see one feeding on a vampire bat in the bottom right. There is occasional arthropod feeding. It's been documented in the literature, but it's not their primary mode of development. So you'll see that sometimes in the colonies. You'll find ones with like green guts where they've probably been feeding on insects that live inside the wood rat nest, for instance. And because they have these specialized diets of mostly blood, they have to have somatic bacteria in their guts that help them get things like essential amino acids. For their host seeking, it's similar to other blood feeding arthropods, but there hasn't been as much work. Like with mosquitoes, we kind of know what distance they send CO2, what distance they start smelling your body odors. Not as well defined in triatomans, but CO2 is probably a long range finding, variable. Then they look at host odors. Animal urine's been shown to be really attractive. As they get closer, you have water vapor in your breath that's been shown to elicit a response, as well as body heat that they can detect. When we think about their movement, the nymphs especially have what's called positive thigmotaxis. So this is like what rats and cockroaches do, where they like to be inside of a crack and be close to walls, and they feel cozy when they're in those tight spaces. The nymphs especially are very cryptic, and they actually have little curved hairs all over their body to hold in dirt and things like that to help them get camouflage. Like this, that center picture on the left is a Triatoma gerstachii on limestone, and it has bits of the dust kind of covering it all over, which helps it blend in perfectly with the limestone rock. They use aggregation pheromones to kind of form these informal colonies. So they, in those refugia, they'll put out chemicals, and so when it gets close to daylight, they'll kind of all retreat back and aggregate together. And when they're not, especially like in an animal nest, the adults will do dispersal flights, and usually this is very seasonal. So for instance, here in Central Texas, there's overlapping generations of the nymphs and the adults. You could find them all year round, but most people encounter them in June, July, and August, when they're actually, the adults fly out, mate, and look for a new animal nest to live in. There's a lot of different environmental factors that affect where you find certain species, but there's one specific kind of gradient I want to talk about, and so that's the sylvatic to domestic environment. So sylvatic is, out in the woods, natural habitats. There's a lot of diversity out there, a lot of animals that they feed on. But as you get closer to human settlements, there's certain species that like these environments, especially in Latin America, and those are the species that are more likely to transmit the disease to humans and encounter humans to pass on Chagas. So I'm not going to go into a ton of detail here in the identification. I mean, it's a little difficult. There's no easy, what we call synapomorphic trait, like one dead trait that I can give you that you're like, okay, that's definitely a kissing bug. Instead, we use a combination of characters. So for kissing bugs, they have a distinct neck. They have the short rostrum, the proboscis. There's a sulcus that they use to make noise on their belly, but you'd have to have a microscope to see that. And they have these two cells on their wings if you have an adult. But then when you go to Triatomanae specifically, the subfamily that has kissing bugs, it's a whole other set of traits. And sometimes it can be difficult for people to tell, if it's a kissing bug or another kind of assassin bug. One of the big characteristics is that they have the proboscis or rostrum is pretty straight, and it's held very close to the body when they're at rest. That's probably one of the quickest giveaways for identifying a triatoman. And for us entomologists, we then move forward to find out, okay, what species is it? And there's a great revision of the whole group that was published in 1979. It's usually your starting point, and then you have to go to more recent literature. And we use things called taxonomic dichotomous keys with a lot of technical jargon and illustrations to identify what species they are. But for a lot of our lay people, or for the other parts of the One Health puzzle out there, I would really point you at some of these new AI mobile apps. So things like Google Lens or the app Seek, which uses data from iNaturalist, are actually pretty effective. Hopefully they won't be putting me out of a job anytime soon, but they're pretty effective at identifying these bugs. So here are some kissing bug lookalikes and some true kissing bugs at the bottom. So a lot of times we're looking at the shape of the head, kind of if they have bright coloration, they're usually not kissing bugs. They usually have pretty thin legs. Here's some more examples. So these are some common US kissing bug lookalikes. So none of these are actual kissing bugs. These three are different kinds of assassin bugs in the Reduviidae, but not blood feeders. And so I know that was a pretty brief look, but I want to give you guys we'll move on and do this quiz here. Again, this is for competition for the Ento coin. So let me pull this up. Bear with me for one. Okay. So if you could take out your mobile device, or you can use a web browser, you can use either that QR code or this link up here in the upper left and put in the game pin. And we'll give you all about 2 minutes here to log in, put your name, and then it's going to grade you both on how quickly you respond, the accuracy, and then how quickly you actually you respond to. So we will, at the end of this, have one winner for that that U.S. Army entomology coin. Oh, we got a bug Jedi in here. All right, people are still coming in. All right, we'll give it about 30 more seconds. Ten seconds. All right, let's start it off. Just to show you the functionality here, we're gonna have a quick poll. This is not gonna count towards your total. Just to show you how it works. The link in the pin down here if you didn't get in earlier. Okay, great. 7% have this on in the background. I appreciate you being involved. All right, is this a kissing bug? Or this is a kissing bug, true or false? I'm gonna give you 10 seconds on these first two, and then it's gonna get faster the next couple ones. All right, false, that was a leaf-footed bug, or the Western conifer seed bug. Skinny human, lead the way. All right. Again, we'll give you 10 seconds on this one. This is the kissing bug, true or false? That was also false. That was a Reduviid from Arizona, not Tritoma. Brad Bear's taking the lead. All right, this is going to be faster. Phew, you guys got it. That was Tritoma protracta. Fort Bliss, actually. Dad takes the lead. Okay. True or false? False. Nice curved proboscis. That was another Reduviid. It's not a Tritoman. Cat's got those quick fingers. False. Yeah, it was a kind of stink bug, a pentatomid. Oh, cat. Crushing it. Ooh, that was Tritoma gerstachii, Texas species. All righty. Cat still in the lead. False. Yep, that was a gorillus, or it's a wheel bug. Another kind of assassin bug. That's still going. Nice. True, that was Rodnius. That kind of kissing bug. Alrighty, Cat. Not sure why this one's got music. False. Yep, that was a milkweed bug. Still going. All right, last one. False. That was Zealous, an assassin bug. All right, let's see. Did anyone dethrone? Cat, ooh, the last one. Dr. Hamer, first place. Let's see. Tom. All right, Tom. Get with me after the end of the talk here, and I'll get your contact info, and we can get this coin to you.
Okay, Major Linhart, just a time check. It's 12:32. So
Roger. Keep moving.
yeah, that was fun though. Thanks.
Okay. All right. So at this point, I'm going to hand it over to Mr. Bast.
All right, I've requested control. I think we've got it. Hope y'all will bear with me. I am dealing with a cold, and so hopefully I can keep things on task here. So let's dive into some of the more practical aspects of Chagas and triatomine bug prevention and management. First, when it comes to military readiness, we really have two vulnerable populations. As Paul mentioned earlier, we've got our human as well as our canine service members. We mentioned both the acute and chronic forms of Chagas, and they each carry their own risks. Our canines are really the population at risk when it comes to the acute disease, which can lead to sudden death. And exposure risk is really also much higher in our canine populations. And that's partly due to their physical exposure, but also because their proclivity to ingest bugs or lick the bite sites. Generally, canines in the DoD that are found to be infected are usually retained in the service as long as they're determined to be fit. Severe morbidity or mortality in adult humans are rare during the acute phase, and the risk is primarily in the chronic phase. One unique aspect that military members might experience is what we call a walking blood bank. Paul mentioned that we do, screen during blood transfusions, but in combat operations, it's possible for battlefield blood donations to occur, and it's really difficult to screen at that point. So it's possible that a chronically infected individual could possibly transmit during one of these events. When possible, rapid screening tests are done, but if not, usually samples are retained from each donor and then sent back for comprehensive testing at a later time. Again, usually medical separation, it's possible or not possible depending on each individual, but that can result in, a loss in good experience for the service. On this map, it shows the distribution of North American triatomines, and it's overlaid with the installations operated by active duty service components. And as you can see, most of our military installations do lie at least within one species range. When talking about surveillance techniques, we have kind of both this active and passive measures. Passive traps are ideal because of the whole set it and forget it concept. But they're not very effective in trapping triatomine bugs. You can use high-powered lights and sheets, but they only capture dispersing adults and only during certain times of the year, as Paul mentioned. And certainly, researchers are working to continue to attempt to make these tools a little bit more useful in routine surveillance. Neotoma wood rats, also known as pack rats, they construct their nests out of whatever is available in their environment. And you'll see a couple of slides, what that can look like. But besides from the wood rat nests, which can be deconstructed kind of a layer at a time, you can have burrows like the one on the bottom left. Those were common in South Texas, and I know our team had done some like soft tick surveillance in these burrows using carbon dioxide as bait, and it wasn't uncommon to see triatomine bug crawling out of the burrow in search of a blood meal. On the East Coast, unfortunately, the mammalian blood hosts are not as well characterized. And I know like folks like Dr. Peterson at University of Delaware are trying to identify, what the blood meal is in the triatamines on the East to target those surveillance efforts a little bit better. But as you can see, wood, rotting wood and other things like that are also good surveillance sites. Here you can see two different wood rats nests. The one on the left is from mountainous part of Fort Bliss in New Mexico. And that's a Triatoma protracta. On the right, you can see a Triatoma rubida nymph from a wood rat's nest at Fort Huachuca in Arizona. On the upper left and right, you can see some folks deconstructing a wood rat's nest. It's labor and time intensive. Usually the wood rats will rebuild their nests after this disturbance has occurred. In the center there, you can see a engorged Triatoma rubida nymph from a wood rat's nest at Laughlin Air Force Base in Texas. You can see in this case all around here, we've got this is made out of cactus pads, and in the middle you see these mesquite beans that are forming the midden of the nest. On the bottom left, we've got Paratriatoma luticularia. That's an adult, and then a nymph on the right. This is a large wood rat's nest, actually just a couple 100 feet away from a military working dog kennel at Fort Riley, Kansas. And then on the right, that's from the Marine Corps Air Station Yuma in New Mexico. That's a wood rat's nest with a Paratriatoma hirsuta nymph. And Paul informed me that this particular nest had a speckled rattlesnake in it. And snakes aren't the only thing to be worried about when doing these rodent nest deconstructions. Respiratory protection might also be necessary if you're like in a haunted virus endemic area. On the top here, you can see these first three images are kind of a good example of how a good kennel should be laid out. You can see here that there's outdoor runs, and the canines can come in and have an indoor space to spend the night with these guillotine-style closing doors to keep them protected. But we do also have some kennels that are a little bit more open and exposed to the elements. This one on the bottom left, you can see there's a lot of lights that can attract bugs and habitat that surrounds it. And these have some sleeping areas, but they're not really closed off from the elements. And then you can see when the dogs are training, particularly in our Texas at Joint Base San Antonio, you can see the handlers will often tie up their dogs to trees. And it's possible that some of these wood snags, have kissing bugs or triatamines that are nesting in them. It's not typical for a kissing bug to come out for a blood meal during the day, but it certainly can happen. A simple way to conduct surveillance is just to leverage certain citizen science sites. You can see here on the left, iNaturalist, which was mentioned by Paul before. There's also a bugguide.net, and excuse me, the Texas A&M University has also an extensive citizen science website that they maintain the records of all the specimens that are coming in from different parts of the United States. Again, the data is for these sites is kind of biased to where a layperson might encounter bugs, again, like mostly dispersing adults attracted to lights. Because kissing bugs primarily host-seek at night, the highest attention should really be paid to preventing kissing bug exposures in sleeping locations. And identifying the high-risk locations will drive the kinds of, excuse me, prevention methods that really should be employed. Buildings with a lot of dilapidated, wooden mud structures or wooden thatched roofs or things that are open on different sides. Anything with animals that are nesting nearby, or when domestic animals are present, again, that urine is going to attract more kissing bugs to the area. The DoD's developed what it calls a DoD insect repellent system. We believe it's really the best strategy for the defense against insects and other arthropod disease vectors. The system includes an application of EPA-approved repellent to exposed skin, permethrin to the field uniform, and then a properly worn uniform. Sorry about that. When high-risk lodging situations identified, then incorporation of a insect repellent, i.e. permethrin, treated bed net is also appropriate. These are the pesticides or the skin repellents that are in place and on the DoD's standardized lists. We have some DEET-based repellents as well as some non-DEET alternatives, such as the picaridin, as well as IR 3535, which are the active ingredients in these two products. As far as clothing goes, most of our uniforms are treated at the factory. The few that are not can be treated with this, what we call a shake and bake kit. That's becoming less common as we get more of our uniforms that, come treated. Sometimes we operate in places where we have, we are authorized wear of civilian clothing. In that case, we have both a pump spray as well as an aerosol version of a permethrin treatment, which is good for 6 weeks or 6 launderings. Besides personal protective measures, there's a number of other IPM strategies or integrated pest management strategies. They can be broken down into cultural, physical, mechanical, biological, and chemical. And for the sake of time, I'm not going to go into all of them. I've covered some of them already. But you can see, like under physical, mechanical, door sweeps is a really good way to ensure kissing bugs can't enter. We don't really have good biological control mechanisms or organisms, if you will. And for IRS sprays or insecticide residual sprays, we don't really have anything that's EPA approved in the United States, although you can use certain products that are registered for certain areas like crack and crevice treatments. You can apply a pyrethroid and it might be effective and not off-label, if you will. We also have some in South America. There are a number of products used, particularly deltamethrin and alpha cypermethrin. But we're seeing resistance occurring in some parts of Latin America. So some of those tools that we have in our toolkit are going away. There's also some pyrethroid impregnated paints that have been used in parts of Latin America. And we're also looking at ways that we can protect our military working dogs, particularly in those open kennels. There in San Antonio, a trial is underway that's looking at pyrethroid impregnated netting material that can surround the kennel and protect them with the bugs from getting in the first place. Primarily, if you apply a perimeter spray and the bugs cross it, especially the non-flying, they can get a lethal dose and then die in the kennel and then get consumed, which is not particularly a good situation. So I think I have left enough time. Maybe Paula, if there's questions, maybe we can address a few questions.
Yeah,
We'll turn
we're
it
at
back over. Sorry, go
we're at
ahead.
12. Yeah, we're at 12:45. So I know you guys have about 10:15 for the case. How about if anybody has any questions, you can put them in the chat and we will field the questions, and then at the end we'll do open dialogue. And if we run over, that would be okay at the very end for open. Does that work for everybody? Thumbs up. Awesome.
Sounds
I'll be
good.
monitoring the chat and I'll interrupt if I have a question I can't answer.
Okay, so like Paul mentioned, this is a part of the presentation where we want some open dialogue, some active engagement with the audience, and so we're going to go over this hypothetical scenario. We are involved in the Joint International Exercise Iron Wood Rat 2025 with a hypothetical nation of Atropia. Which will take place in endemic areas with Chagas disease transmission. And as part of the pre-operational medical planning, you're briefed about various parts of the operation, which I will do now. So some of the things we want to get out of this, we want to be able to identify high-risk areas and activities, what kissing bug surveillance, if any, you would do. What pest management tools would you use and where? And how would you communicate those risks to the involved U.S. units? So this is our operational area. Starting on the right, we have a team house in the town area with the special operations team. We have a veterinary clinic where we're doing local training of local vets. There's a small arms training range in a somewhat open area bordered by some forested area. And then we have a military working dog kennel with chain link fences, local mascot animals also in the area. We've got a troop lodging at an old barracks. We have an opposition force, which in the military is like the bad guys, if you will, that are designed to attack or go against the good guys, and they are sleeping in the woods overnight in preparation for an ambush. And then we have troop lodging tents in an open field and troops using an old barn as an observation post. So we will go and look at some of these locations. Here we have our kennel with the open chain link fences. That was located right here. We have troop lodging at Old Barracks, which is right here. We've got the troop lodging in the tents in the open field, which is located right here. And then we have a special operations team house, and that was located over here. Then the second slide, here's the opposition force sleeping out in the field in prep for an ambush right here. Veterinary training at a local clinic, which is happening right here in the town. Small arms training, which is occurring right here in this field next to the wooded area. And then troops using an old barn as an observation post, which is located right here. So I want you guys to think about those areas and identify the high, medium, and low risk areas. And Paul is going to take it from here and bring in a poll for you to answer those things.
All right, everyone. So we're going to quickly use Kahoot again. There's no prize for this one, but we want to let everyone kind of, because we have such a large group, quickly give their 2 cents about where they think the different risk levels are. So if you could follow this link again, use the QR code, give you another 45 seconds, kind of get in here. If you missed this initial push, there's a link and the pin at the bottom so you can still join. Don't worry, there's no wrong answers here. We have kind of our ideas, but you know, everyone can kind of look at the situation a little bit differently. All right, I'm going to go ahead and get started here. Okay, so we'll start with the military working dog kennel, that chain link cages, and they also are keeping some local mascot animals there, something that's usually not allowed in the United States military. I'm sorry for my green suiters out here that we couldn't get these things color coded to be, you know, a stoplight. So you just have to remember that blue is medium risk and yellow is low risk. Okay, high risk. Yeah, I would agree with that. As we talked about earlier, very easy routes for the dogs to get exposed there. Okay, we got troops lodging in an old barracks. So possibly dilapidated buildings. You know, we don't know what the lighting situation is. We don't know if they're air conditioned and kind of closed off, but just judging from the pictures, they're old. There's possibilities of those kind of routes of entry. Okay, we're a little split here. I would probably classify this as medium risk, definitely worth the look to see if there's, especially if there's an active infestation or colony in some of these buildings, then it would move to high risk, but yeah, I would put it medium. Okay, we got troops lodging now in an open field. So we've got pretty new looking army tent here. Looks like they have AC going, so there's positive pressure in there. We're an open field. If you saw the map earlier, it was pretty far to the wood line. I'd say probably a couple 100 yards. Where do we fall here? Okay, well, we have a lot of people saying high risk. I would probably also put this one at medium risk as well. Again, there's possible routes of entry there, but there's some, in terms of the other events we talked about, there's some higher risk things out there for sure. All right, now we got special operations. They're the cool guys operating out of their team house back in town. This looks like a pretty upper middle class neighborhood in Atropia here. Pretty new construction, AC, looks like good quality doors and windows. So where do we classify this? Low risk. Yep, they're probably pretty safe. At least when they're staying in the house, they might go and do some cool guy stuff in the woods, but when they're sleeping there, they should be fine. Okay, opposition forces. They're bivouacking in the woods overnight in preparation for an ambush. Joe's just sleeping here. He made a fighting position out of rocks. He's got his woobie. That's a army slang for that blanket. He's out in the open, and it's natural habitat. We've been saying this is an endemic area. There's probably plenty of wildlife nearby. High risk, yes. All right, the vets are working with local nationals. They're training in a local clinic. They're working with either the local nationals, working dogs, or some privately owned animals. What do we think here? Okay, yep, low risk. Especially as long as they're wearing gloves. A lot of those vets weren't in that picture, but if they're dealing with any blood or fluid, I would definitely want to be wearing gloves. But otherwise, yeah, low risk. All right, small arms training. We're working with local nationals. We're using the range during the day. We might have some soldiers kind of resting in the background up against buildings and things like that, but mainly middle of the day out using the weapons range. Low risk. Yeah, I'd say this is pretty low risk. Again, unless you got some soldiers just sleeping in a dilapidated building in the background, probably very low risk of transmission here. Okay, finally, troops using an old barracks as an observation post. I've known some people have deployed, have seen soldiers doing this and done it themselves. Obviously, when you're in a mission, you have to use the terrain and structures that are available. But what do we think about this building and them staying in here, 24 hours a day? High risk, yes. Okay, great job, everybody. So let's go through these other deliverables really quick. And here, I'm going to ask if, with our remaining time, people can raise their hands if they've got a comment here. Let's see, I'm going to open my camera now. Okay. All right, so what kissing bug surveillance, if any, would you do? And I'm going to bring up the map here with kind of our classification of those different events. If somebody could raise a hand and kind of tell me what kind of surveillance, based on what we've talked about today, would you do during this operation? Any volunteers? Anyone be brave? What's the easiest one we talked about? So there were some websites, right? So let's say, you're going to this location, that of surveillance you should do. I talked a little bit about it during the poll, based on some of those buildings. So kind of, how would you determine if some of those things are medium or high risk? Oh, Dr. Casey.
My mic, can you hear me?
Ma'am, yes, I can hear you now.
You could do, in addition to your, before you go, you could do your literature search or CDC or WHO, wherever, in terms of what the species are there. But the target areas for actually a walk around and looking would be the kennel, the troop lodging, do a walk through that area. Are there, do you have any habitat shrubs or old brush in the area around the barracks? And certainly, probably the highest ones would be the troops using the barn because of the shape of those structures. And then doing some surveillance to see if you've got wood rat nest or other likely burrows in the bivouac area.
Thank you, ma'am. Appreciate the comments. Yeah, definitely. I'm full agreement. Probably the kennel first. So check that out. Then those buildings and the tents. Look in the local vegetation and see what the habitat's like there. Are you seeing a lot of animal nests? Definitely. So that, again, that might be labor intensive, and but depending on the operation, you might have to balance that with whatever other threats you're working with. As preventive medicine officers, sometimes we have to be doing food and water inspections and air sampling and a lot of other things. So definitely. Can anyone give me suggestions for any kind of passive surveillance we might want to try that might be less labor intensive? Yep. Oh, we got a hand.
Are you suggesting, or the first thing that came to my mind was setting traps?
Yeah, so right, there are some traps in development, but we don't have any generally accepted ones. But if you procured a UV light or mercury light, a lot of us entomologists have those. You could definitely use that. And then you could also just walk around the building lights at night in the installation there and see if you find attraction to that. So you can kind of co-opt the infrastructure that's already there. So appreciate the comments. All right, so Josh went through the pest management things. So obviously we're strapped for time a little quickly. And you heard about a lot of tools that don't work. But what kind of pest management tools would you at least try? Let's say your surveillance did find quite a few kissing bugs. what kind of actual pest management practices do you think you would use here? And for our people that are focused more on, installation stuff here in the United States, you could think about that, not so much a military operation overseas, but here as well. Yep. David, you got another comment?
I would spray.
Spray.
Preventive.
Okay.
Preventive.
Yeah, if you're finding them, especially in your high risk areas, you spray. It might be a little tricky depending on what the operation is like. You have to work with your local nationals. If you're here in the United States, you could, and especially if you're in a state that allows site treatment, you could do those like crack and crevice sprays. But we really wanted to push one of the prevention measures earlier. It's kind of a mechanical barrier. So what do you think you would advise a lot of the troops on this operation to use? And really, you probably need to get ahead of the operation before they even went to make sure that they had these in their inventory. Anyone kind of chime in here? Oh, yep. I'm seeing netting in the chat. I'm seeing netting for sleeping. Yeah, bed nets. Yeah, that'll be your number one of the tools that are still effective. That mechanical barrier, if it's impregnated with permethrin, all the better. Right now, we don't have a good technique or product for protecting the military working dogs, but if you can co-opt that netting as well for those military working dogs in this situation, all the better. That's really your best toolkit, or best tool in the kit that we have for doing this. The other one I'd really point out is education. So you need to do those briefs to your soldiers kind of before you're going. And we'll talk about that for the last deliverable here. It's really important to get ahead of these operations and kind of communicate the risk. You want to you have to balance it and not overstate that risk, because they're going to be facing a lot of other things in an operation like this. You'd want to, let's say dengue's in the environment. You really want to hammer that home above Chagas, to be honest. Or foodborne disease outbreaks, but they still need to be aware that this is something that could, if they get infected, obviously kill them decades down the road. So education is one of our best tools as well right now, and I hope that we've kind of given you a lot of those tools today. So with that, I'm going to put on the screen here some reach back support options. So for our military folks, we have the Armed Forces Pest Management Board, and this is across all the services. There's a lot of resources up there for how stuff we covered about the uniform and other products, where to the NSNs for ordering bed nets, etc. are up there. And there's a contact link right there for both people that are within the United States and that are deployed overseas. And there's resources there that you can contact. We also have the Defense Centers of Public Health Aberdeen that Josh Bast is part of. So there's a contact link there, as well as a fact sheet that they produced on Chagas. And then if you're in our Western region for the Army specifically, so we're from the Mississippi River to California, my civilian counterpart, Mr. Walter Rochelle, there's his email as well, and he's got a lot of experience. He's been sampling kissing bugs and working and advising installations, through my time with the unit, through Josh's time with the unit and before. So he's a wealth of knowledge and a good person to be in contact with if you're in the region. And again, we want to thank Paula for hosting us, and we hope you all have taken some good information away from today.
All right, thank you, Major Linhart. Thank you, Josh. If you have some more questions, feel free to put them in the chat. I have one quick announcement. You can scan this QR code or click the link in the chat. Please take our evaluation. If you do need CME credits, we're happy to give them. They are free. And just fill out your evaluation, let us know you need them, and we will be happy to send them to you in a couple of weeks. We'll email them out to you. Next slide, please. And then lastly, and thank you all I know we're a few minutes over, but I think we did really good timing, cramming everything in here. We will have this recording posted on our website. We've also asked if we could share it in other places, and we're going to work on doing that. Happy to share this with others, spread the word. And then if you have any questions, you can reach out. My email address or the chagasus@gmail.com. As I mentioned, we'll put everybody who's here on the list for the next one. The next session will be August 22nd. Major Joseph Marcus, infectious disease specialist at the Brooks Army Medical Center. Is going to be talking about Chagas disease presenting in blood donor screenings through the military. It's going to be fascinating. This is one of the best sessions I think I've ever attended for our ECHO sessions. Is a lot of fun. So thank you for both of you for your time. And I also want to thank our team, UT Health, San Antonio ECHO, as well as my research assistants and Dr. Zhu from Texas State. And if you have any questions, we'll stick around for a few more minutes. Happy to chat. Thank you for coming. Hope you keep your lookout for the kissing bugs wherever you are. Oh, there's one right there. We've got one in a cup. I keep one in a glass jar to show to my students on a regular basis. We call him Dead Fred. It's actually one that my students brought to me. Look at that. You've got one right there too. Awesome. Thank you all for being
Thanks for
here.
having us.